Hello, we are students from Harmony School of Excellence, and our project is about the glowing sea, which is actually just bioluminescence.
The science portion of our project consists of bioluminescence, which is the production and release of light by a living organism. The light is created when a molecule called luciferin is catalyzed by the enzyme luciferase, producing light as a byproduct. This process requires energy, typically in the form of ATP from sunlight through photosynthesis, and results in a glow that can vary in color, depending on the organism. In this case, dinoflagellates emit cold light. That is why it's a blue color, making it more captivating to the eye. In addition to the products of their photosynthesis, they produce sugars of self-sustainability, and as a whole produce around 50% of the oxygen of Earth. Along with that, their bright glow isn't their only way of presenting themselves. They also cause red tides as a result of overpopulating, scientifically referred to as harmful algae blooms.
Further on, our social studies component is that dinoflagellates have had a long history. They've been a wonderful phenomenon for centuries, assumed to date back to biblical times, and have come as far as today. For example, the red tides were logged by explorers of the 17th century, interpreting the event as bad omens. And even Christopher Columbus, along with other sailors, got to see the golden seas, describing it in the logs as burning waters. However, it wasn't until the 19th century that scientists began recognizing the effects of the red tides, which were identified in the 20th century as a result of the advancing microscopy used to study marine plankton more closely. And then governments started creating programs for scientific study in order to mitigate the harmful effects of algae blooms caused by the dinoflagellates, as they were provoking coastal economies to have troubles. But as of today, there has been a rise in ecotourism of countries like Puerto Rico and Jamaica for their unique shores that glow.
For our project, we modified the day and night cycle of dinoflagellates to investigate how light exposure impacts their bioluminescence. We divided the culture into 6 colonies across 3 experimental conditions. One group experienced constant light all day environment, and another kept in constant darkness all night environment. And the third group followed an inverted day-night cycle to simulate night during the daylight hours for easier observation. Our goal was to understand the environmental and biological factors that influence the intensity of their bioluminescence. Our findings indicated that light exposure and circadian rhythm significantly affect the dinoflagellate's ability to glow. The group with the inverted day-night cycle, simulating regular day and night patterns, exhibited the brightest and most consistent bioluminescence. In contrast, the constant light group showed diminishing brightness over time, likely due to overstimulation, while the constant dark group showed no bioluminescence at all. This experiment suggests that both the duration and timing of light exposure are crucial for sustaining optimal bioluminescent intensity, as the dinoflagellates require specific light-dark rhythms to regulate photosynthesis effectively. Here we can be seen with our beaker and our dinoflagellates. We are going to be pouring in our dinoflagellates, and first we made sure to clean our beaker to make sure that we did not contaminate our experiment. Here it is, all rinsed out. And then I, Brandy, will be pouring in the dinoflagellates with the help of Genesis, holding the beaker to make sure it doesn't fall. We have a really pretty flow right here. Once we are done pouring the dinoflagellates in, we are going to feed them, and we're going to pour the nutrients inside of the dinoflagellate bag to make sure that we get any remaining dinoflagellates. We are going to be mixing the nutrients around with the dinoflagellates here. If you look closely enough, you can see the little dinoflagellates swimming around. They do look kind of yellow. They're like little spots, like dust. Thank you for listening. We hope you got to learn more about this fascinating topic and some of the properties that we got to experiment on these living organisms.
If you have any more questions, please refer back to our site, The Glowing Sea.